Okay, thank you everyone. Thank you for the opportunity to be involved in this event community and having the opportunity to participate and helping to organize the symposium. Today, I'm going to talk to you about some really preliminary work and some ideas that we're doing that's coming out of my research lab at UC Merced. I started in the middle of the pandemic, so things are kind of slowly going, but going nonetheless. So not only are we going to go back into the marine world, we're actually going to go back into coral reefs because I think these ecosystems are just so iconic and so inspiring in terms of thinking about microbial interactions in the sea. We know that they're really important ecosystems to world economies. They contribute upwards of like $400 billion per year to global markets, and so they're super important. And what I've always been super fascinated about is the actual microbial interactions that are occurring on these reef systems. So I'm interested in all the different types of microbes. I haven't looked into the apocomplexans yet, but I know I'm pretty inspired after listening to Patrick's talk. Thinking about how these different microorganisms are actually producing organic molecules to feed one another and to help support host metabolism and health. And one of the reasons why I think this is super important to study is because, of course, time is running out. We know that these reefs are becoming bleached, they're losing their algal symbionts, and that's leading to decline in reef ecosystems worldwide. Of course, probably all heard that there's a desperate push right now in the Florida Keys itself, where we know that they've experienced an incredible amount of high temperature events that's leading to drastic changes in reef ecosystems. So where I think that we can make a contribution, though, as microbiologists, is thinking about how we can use microbes to actually help mitigate some of these damages. So there's been evidence now suggesting from several different groups that we can engineer bacterial communities to actually help protect corals from stress itself. And so the idea that we can design probiotics to actually help mitigate the impacts of thermal stress. So I think that these experiments have been really cool. They've been showing that they're working. And what I'm interested in sort of moving towards is actually understanding the mechanisms behind these interactions. So how do these microbial communities work together within these complex communities to actually benefit the metabolism of the host? And to do this, because I live in Merced and not in a tropical reef ecosystem, we actually started working with a sea anemone called Aptasia. It is a common model system that people have used in the lab because it's really easy to grow. We can actually cut it in half and it'll grow up to individuals. We can manipulate the symbiotic composition. And it has, it has a really interesting, or has the same symbiosis as coral reefs do. So we know that it has these symbiotic partners. We can remove them through cold shock or through heat stress. And it has a complex microbial community, the function of which we don't really know. So we know that there are many different partners that are persistent in this Aptasia individual. So while a lot of the work in Aptasia so far to date has really focused on thinking about the role of the symbiotic and the cell and molecular biology behind those interactions, what I'm really interested in exploring is how does the Aptasia microbiome help support host health and metabolism? So what we're currently doing is taking a classic microbial ecology approach. We've been looking at our clonal lineages of the Aptasia. So again, we just have a couple different strains. We can split them in half. We can study their microbial dynamics. We've been doing some longitudinal studies and really just at first looking at the 16S rRNA sequences to get a sense of how does the community changing over time. And what we know is that there are about 50 core members within this microbial community, and they span many different phyla and taxonomic divisions. What is concerning to me is if I want to study how these microbial interactions are working, how can we study it in such a complex community? So we decided to start to try to actually manipulate the composition of the microbiome. We've been trying to simplify the microbial community through the addition of antibiotics, and this is work that Sophie, as a PhD student in my lab, is now currently pursuing. And we want to see if we can actually disrupt the symbiosis that is present within the microbiome itself. So we started doing this as an antibiotic disturbance and recovery experiment. It's kind of how we started thinking about it, where we first held these anemones in filtered artificial seawater to try to reduce the microbial load. And then we treated them with different types of antibiotics and monitored their communities over time. And we also monitored the recovery of the microbial communities after that antibiotic disturbance. So of course, what is really promising is that we can overall reduce the microbial load of these anemones with antibiotic treatments. The pink circles are always going to be my treated anemones. The black ones are our control groups. And we can keep them actually reduced about a week after the antibiotic exposure in terms of the absolute load of the community composition, but they bounce back after 3 weeks of recovery in filtered artificial seawater. It's not surprisingly it's not very surprising. When we treat our own guts with antibiotics, they'll bounce back again after a short amount of time. And of course, what is interesting though is what's happening on the community level in terms of changes. So we did a number of alpha diversity and beta diversity statistics to try to get a sense of what is happening. So during the antibiotic exposure, if we look at just the number of ASVs initially, we don't really see a lot of changes in the absolute number of ASVs until the recovery period of the experiment itself. So the composition of the community isn't really changing when we're introducing the antibiotics, even though the microbial load is decreased. The second thing that we see in our beta diversity statistic, which is the bottom set of plots, is that we see that the beta diversity during the recovery is significantly reduced during that recovery period. So to me, this says two things. Our community is less diverse after 3 weeks post-antibiotic exposure, and to beta dispersion values, we see that as the lowest after 3 weeks post-antibiotic exposure. And this is reflected again in that multivariate statistical plot, where we see that the lowest or the tightest forming group is at that 3 weeks of recovery in filtered artificial seawater after the anemone has been exposed to antibiotics. So overall, we can conclude that our antibiotic treatments were effective. So the second thing that we see, or the third piece of evidence I want to sort of point out, is that when we use the antibiotic treatments, we're actually able to remove about 58% of the core community at the final recovery time point. So we don't see it being removed during the antibiotic exposure, but again, during that recovery period, we see that the community is largely reduced in terms of its composition, and that's displayed by these bubble plots and showing relative abundance values. So I wanted to think about, and I've been thinking about how we can explain some of these data and what does it mean in terms of when we think about recovery dynamics of microbial communities within these microbiomes. What we have observed is that there's a less diverse but more stable community following antibiotic exposure. And we see this really nicely in our bar plots where our control samples look very different in terms of the community composition as samples taken at the recovery after antibiotic exposure. And when we think about this less diverse, more stable community, we can kind of picture it something like this. And anemones are just held in normal filtered or artificial seawater media. We see that the community is relatively diverse between individual anemones itself, even though it's the same colonial lineage. But when we expose them to antibiotics, we can actually have a more defined, better, more consistent community between individuals itself. So when we consider what this means for the future of notobiotic strains, it kind of means two different things to us. First, we have the potential to potentially treat with different types of antibiotic solutions, or we can have exposure periods where we treat with one type of antibiotic followed by a second type of antibiotic, which may lead to further reduction in the actual diversity and community composition of the community. This is nice for us because one of the things that we've noticed is that when we actually treat our anemones with antibiotics, they don't look very nice. But when we have these different types of antibiotics that we're treating them with, we see that the anemones look a little bit healthier than they would just under one type of antibiotic itself. The second thing that we're excited about is the closing of the life cycle of the anemone, which was recently put out in a bioRxiv paper. And what is really hopeful to me is that if we can take that closed life cycle and treat those newly settled individuals with antibiotics, maybe we can actually reduce the microbiome and get towards that notobiotic strain. And the reason why I think this is all very important because it'll help facilitate some of the research that we have funded in the lab, thinking about how we can recolonize different strains of Aiptasia with different combinations of microbial symbionts to, again, effectively understand the metabolism and interactions of these bacterial communities inside host organisms themselves. So the other thing that I want to think about, though, when we think about less diverse and more stable communities is what does this mean in terms of alternate stable states? So in a classic ecology, we think of what we're seeing as being a transition between having a very variable community that has a lot of flexibility with it between individuals to a community that's actually much more consistent. And this was really, in our study, facilitated by the introduction of antibiotics, so it's an antibiotic exposure or disturbance event. And what does this mean in terms of subsequent stressors? Well, in human microbiome studies, we know that the introduction of antibiotics can have incomplete, it can have very variable impacts on the human microbiome within our guts. Oftentimes, these microbial communities, after disrupted by antibiotics, is incomplete recovery. We know that there's highly individualized responses, and the resilience of subsequent stress is often very different depending on what type of system you're looking at. So with Anaptasia, what does that resilience mean? If you have these two different types of microbial communities, some that are very flexible and some that are very resistant, and what types of resilient things are we interested in? We're interested in, of course, what are the impacts of climate change on these different types of microbial symbioses? And we want to understand how does that climate change benefit these partnerships or impact these beneficial partnerships between these animals and microbes. So this is going to be the last slide that I'm going to leave with you today. We have a large BII grant from the NSF to study the impacts of climate change across different types of microbial systems, with the idea if we can understand what climate change is doing to these different microbial systems across different animal symbiosis types, and maybe we can help understand or better predict what will happen in terms of global biodiversity loss in the future. So I would like to thank my small lab. This work was largely led by Sophie, who's a now PhD student, and she had 2 undergrads in the lab helping her out. I'll take any questions.
Hi, great talk. So I was wondering, do you see any shifts in functional groups based on your reduced community after antibiotic treatments?
So we know, so I can tell you on the taxonomic level, we have not done any metagenomic sequencing. It's definitely on like the next steps of things that we're pursuing in the lab. We see that several other studies in cnidarians that are treated with antibiotics, we see a reduction in gamma proteobacteria and alpha proteobacteria groups. We can talk more.
So I know you can't go into full depths of your climate change work, but I'm wondering, are you just gonna like ramp up the heat in a bunch of them and just see how they survive, or like what's your plan for that kind of research?
Yeah, so we have 2 sets of experiments planned. Right now, we're working on thinking about acclimatization experiments. So how well does the anemones actually acclimatize to different types of temperature stress, and how long does that acclimatization work, or how long do we need to acclimatize them for? We have 4 different temperature treatments that we're going to look at. So we're going to look at a range of temperature conditions within our anemones. We're also planning on some long-term evolutionary ecology or evolutionary experiments to also think about how these different generations of bacteria are impacting the host over time.
Okay. Any other questions?
Yeah.
So I know they've been treating the Florida reef truck with amoxicillin to fight stony coral tissue loss disease. Have you looked into like the results of that? If the antibiotic treated corals out there have been less resistant to the bleaching that's happening right now?
I don't know if people have, I don't know if there's studies that look at that specific question. I need to look at a little bit more. I know people have thinking about we've most been looking at multiple antibiotics at once, trying to just get as much knocked down as possible as opposed to one type of antibiotic. But that's, of course, one of the questions that I have is if we're treating these diseased corals with antibiotics, is that affecting other resilience parameters of the coral itself?
We have one question online. Dina Beatty says, great talk. Just curious, if you add microbes to a closed system without antibiotics, do you see a change in the microbial community? And also, why do you suspect the delayed impact of antibiotics on the microbial community?
So I'll answer the first question first. So we know that if we change the environment of the Aiptasia host in terms of like the seawater, that the microbiome will vary very differently. So this is a largely transient community that's impacted by the actual seawater that you're keeping in. In terms of the second, sorry, you can remind me the second question.
Right, I'm trying to find it again. Just a second.
It was about antibiotics.
The second part was, why do you suspect the delayed impact of antibiotics on the microbial community?
Yeah. So we know that the microbial community, the overall load of the microbiome of the aptasia is reduced with the antibiotics. And so the composition hasn't changed. So that means that after the pressure of the antibiotics has been released, I suspect that it is being altered it's very variable the week after the antibiotic exposure. And so I'm guessing that different types of bacteria are able to recolonize more quickly than others. And then multiple weeks after that period, it becomes more stable. And so you're stabilizing it onto that sort of composition. About 3 weeks. I hope that makes sense. I'm still thinking and toying around with some ideas in microbial ecology and thinking about this data set.
Okay, thank you so much. It was a great
Thanks.
talk.